Today we will talk about tardigrade, a fascinating microscopic animal. Tardigrades, known colloquially as water bears or moss piglets, are a phylum of eight-legged segmented micro-animals. They were first described by the German zoologist Johann August Ephraim Goes in 1773, who called them kleiner Wasserbar, means little water bear. In 1777, the Italian biologist Lazzaro Spallanzani named them tardigrada, which means slow steppers. They have been found in diverse regions of Earth's biosphere: mountaintops, the deep sea, tropical rainforests, and the Antarctic. Tardigrades are among the most resilient animals known, with individual species able to survive extreme conditions, such as exposure to extreme temperatures, extreme pressures both high and low, air deprivation, radiation, dehydration, and starvation that would quickly kill most other known forms of life. Tardigrades have even survived exposure to outer space. There are about 1,300 known species in the phylum Tardigrada, a part of the superphylum Ecdysozoa consisting of animals that grow by ecdysis such as arthropods and nematodes. The earliest known true members of the group are known from Cretaceous 145 to 66 million years ago amber, found in North America, but are essentially modern forms, and therefore likely have a significantly earlier origin, as they diverged from their closest relatives in the Cambrian, over 500 million years ago. Tardigrades are the first known animal to survive after exposure to outer space. In September 2007, dehydrated tardigrades were taken into low Earth orbit on the PHOTONM-3 mission carrying the BIOPAN astrobiology payload. For 10 days, groups of tardigrades, some of them previously dehydrated, some of them not, were exposed to the hard vacuum of outer space or vacuum and solar UV radiation. Back on Earth, more than 68% of the subjects protected from solar UV radiation were reanimated within 30 minutes following rehydration. Although subsequent mortality was high, many of these produced viable embryos. In contrast, hydrated samples exposed to the combined effect of vacuum and full solar UV radiation had significantly reduced survival, with only 3 subjects of Milnesium tardigradum surviving. Also, it was found that the space vacuum did not have a significant effect on egg laying in either R. coronifer or M. tardigradum. However, M. tardigradum exposed to UV radiation had a lower egg laying rate. In May 2011, Italian scientists sent tardigrades on board the International Space Station along with extremophiles on STS-134, the final flight of Space Shuttle Endeavour. Their conclusion was that microgravity and cosmic radiation did not significantly affect survival of tardigrades in flight, and stated that tardigrades represent a useful animal for space research. In November 2011, they were among the organisms to be sent by the U.S.-based Planetary Society on the Russian Phobos Grunt mission's Living Interplanetary Flight experiment. However, the launch failed. In August 2019, scientists reported that a capsule containing tardigrades in a cryptobiotic state may have survived for a while on the Moon after the April 2019 crash landing of Bereshit, a failed Israeli lunar lander, but in May 2021 it was reported that they were unlikely to have survived the impact. In recent years, there has also been increased speculation regarding tardigrades' ability to survive on Mars without any life support systems. But despite the hardiness of the tardigrade, it would still need stuff to eat in order to set up shop. Tardigrades are usually about 0.5 mm (0.020) in long when fully grown. They are short and plump, with 4 pairs of legs, each ending in claws, usually 4 to 8, or suction discs. Tardigrades are prevalent in mosses and lichens and feed on plant cells, algae, and small invertebrates. When collected, they may be viewed under any microscope, making them accessible to students and amateur scientists. Ecology and life history. Most tardigrades are phytophagous plant eaters or bacteriophagous bacteria eaters, but some are carnivorous to the extent that they eat smaller species of tardigrades, e.g., Milnesium tardigradum. Tardigrades share morphological characteristics with many species that differ largely by class. Biologists have a difficult time finding verification among tardigrade species because of this relationship. These animals are most closely related to the early evolution of arthropods. Tardigrade fossils go as far back as the Cretaceous period in North America. Tardigrades are considered cosmopolitan and can be located in regions all over the world. The eggs and cysts of tardigrades are so durable that they can be carried great distances on the feet of other animals. Tardigrades have survived all 5 recognized mass extinctions due to their plethora of survival characteristics, including the ability to survive conditions that would be fatal to almost all other animals. The lifespan of tardigrades ranges from 3 to 4 months for some species, up to 2 years for other species, not counting their time in dormant states. Physiology. Scientists have reported tardigrades in hot springs on top of the Himalayas to the deep sea 4 meters, 13 feet, and from the polar regions to the equator, under layers of solid ice, and in ocean sediments. Many species can be found in milder environments such as lakes, ponds, and meadows, while others can be found in stone walls and roofs. Tardigrades are most common in moist environments, but can stay active wherever they can retain at least some moisture. Tardigrades are thought to be able to survive even complete global mass extinction events caused by astrophysical events, such as gamma-ray bursts or large meteorite impacts. Some of them can withstand extremely cold temperatures down to 0.01 Kelvin 460 deg F, 273 degrees Celsius, close to absolute zero, while others can withstand extremely hot temperatures up to 420 Kelvin 300 deg F, 150 degrees Celsius for several minutes. Pressures about 6 times greater than those found in the deepest ocean trenches. Ionizing radiation at doses hundreds of times higher than the lethal dose for a human, and the vacuum of outer space. Tardigrades that live in harsh conditions undergo an annual process of cyclomorphosis, allowing for survival in sub-zero temperatures. They are not considered extremophilic because they are not adapted to exploit these conditions, only to endure them. This means that their chances of dying increase the longer they are exposed to the extreme environments, whereas true extremophiles thrive in a physically or geochemically extreme environment that would harm most other organisms. Tardigrades are one of the few groups of species that are capable of suspending their metabolism. While in this state, their metabolism lowers to less than 0.01% of normal, and their water content can drop to 1% of normal, and they can go without food or water for more than 30 years, only to later rehydrate, forage, and reproduce. Many species of tardigrade can survive in a dehydrated state up to 5 years, or longer in exceptional cases. Depending on the environment, they may enter this state via anhydrobiosis, cryobiosis, osmobiosis, or anaxibiosis. Their ability to remain desiccated for such long periods of time was thought to be dependent on high levels of the non-reducing disaccharide trehalose, which is commonly seen in other organisms that survive desiccation, and tardigrades have trehalase genes. However, it has been seen that in both tardigrades and bdelloid rotifers, there is only a partial capability to synthesize trehalose in quantities that may contribute to desiccation tolerance. In response to this finding, more research was done on how these animals survived such extreme conditions. It was found that intrinsically disordered proteins were highly expressed in response to desiccation in tardigrades. Additionally, 3 new IDPs were found to be specific to tardigrades and coined tardigrade-specific proteins. These TDPs may maintain the structure of membranes by associating with the polar heads of the phospholipids bilayers, avoiding structural damage upon rehydration. Also, TDPs, being highly hydrophilic, are thought to be involved in a vitrification mechanism, where a glass-like matrix forms within cells to protect the cellular contents upon desiccation. Their DNA is further protected from radiation by a protein called D-submine, short for damage suppressor. In this cryptobiotic state, the tardigrade is known as a ton. Thank you for joining this journey with us, and thanks to those wonderful people who are supporting this channel as we explore the unseen world. If you want to see more from our master of microscope, then like, share, subscribe, and click the bell icon.
Thanks for watching.